I'd like to welcome everybody to the Plant Phenome Journal webinar for today, Monday, November 2nd. This is based on a paper that Dr. Robert Bruce and Dr. John Sulik published called Plot Extraction from Aerial Imagery, a Precision Agriculture Approach. Dr. Robert Bruce is a researcher at the University of Guelph, and he got his PhD at the University of Guelph, more on the plant breeding and genetics side. Dr. John Sulik is an assistant professor at the University of Guelph, and he got his PhD in geography at Florida State. So he's a US transplant to Guelph. So they'll give their presentation. Afterwards, I'll open it up for questions. The chat, I believe, is open. Feel free to ask any questions there, and I'll read them aloud. I'll also open it up for people to speak at the end if you want to ask your question orally. And if you have any problems or any issues, go ahead and email me or send me a chat. So without any further ado, I believe Dr. Robert Bruce will be our presenter today.
Okay, thank you so much for that introduction, Seth. Welcome everyone to the presentation. So as mentioned, the paper was titled Plot Extraction from Aerial Imagery, a Precision Agriculture Approach. So myself, I'm on the paper, as well as my PhD supervisor, Dr. Istvan Raichen, who's a soybean breeder and geneticist here at the University of Guelph, and John Sulik as well, who was introduced. So we are all members of the Department of Plant Agriculture at the University of Guelph in Guelph, Ontario, Canada. So I'll give a brief outline. So we'll go over a little introduction of why we came up with this method. I'll give a description of the method, as well as demonstrate a couple of the steps of why you might want to use this and how you might go about doing that. I'll show some final results of our data layers on top of each other, so you can see how it might benefit your own research. And I'll end with just a couple slides on how we've actually used this method already to speed up some of our research objectives. So as an introduction, researchers are increasingly using aerial images for a lot of purposes. And in some cases, from people I've talked to, they're imaging fields and they don't actually really know what to do with those fields, but they know that they can do that. So a major bottleneck remains in handling a lot of this image data. So you image a large field that might have a couple thousand plots in it, but how do you actually translate that large image data set into a categorized plot data point quickly and efficiently? So there are kind of three current classes of methods that we use to do this type of work right now. So those would be kind of a manual delineation. And that's where you go in and draw those plot boxes by hand. This is very time consuming, and it is really not scalable. Once you get up past 100 or 200 plots, it's not a very good use of resources. There are machine learning or computer vision methods, and these, would use some kind of algorithm to attempt to outline the plots. This is a bit of an exaggeration, but as you can see, like when your plots kind of bleed together, you may not get the best separation of your plots, and this might depend on the time of year of your image or your plot shape or your field conditions. And as well, there's a number of methods of systematic spacing. That's where you apply some kind of grid over your field. This works best with a very uniform field. But if you have maybe a bit of a crooked field, or you didn't have a precision planter when you were planting, you might have to go in and do some manual corrections to really get a good plot extraction and good spacing. So the kind of general overview of our method is a precision ag approach. So the idea is you take a georeferenced drone image. So this would be kind of a centimeter level accurate georeferenced drone image. You overlay precision planter data for your plot trips, and this would be a direct output from a precision planting implement, whether it's self-propelled or something you tow along. And then you can generate your plot boundaries directly from this planter data. And I'll describe a little bit more about all these steps as we go on. So the general model workflow is you have a centimeter level accurate plot position data and a centimeter level accurate georeferenced image, whether that's aerial or from a drone or from a satellite. Briefly recap, so a major bottleneck really remains getting useful data out of these large image data sets. So as I mentioned, the kind of three main types of methods, you have your manual delineation, that's where you get your boxes by hand, computer vision type approach, and as well as a systematic spacing approach. And they all have some level of effectiveness, but they also have pretty big weaknesses depending on your images and the fields that you're working with. So the proposed method, like I mentioned, is you take your drone image, you overlay your planter data, and you can very easily generate your plot boundaries for your downstream analysis. Okay, so you overlay your data. So yeah, the generalized workflow. So you have your plot data, you have your georeferenced image, you can overlay them. Then we can convert our plot points out of our precision equipment to polygons using the QGIS rectangles, ovals, and diamonds tool. We use QGIS for all of our GIS work. We can assign our plot IDs using a field plan, and there's a few ways to do this, and I'll describe that a little bit later. And then from that point, we can extract our plot zonal statistics for downstream analysis, whatever that might be. So a couple of the details of what we did in the paper. So our image acquisition was done mostly with the DJI M210 with a MicaSense Red Edge camera. We used a grid flight pattern with 80% overlap. And importantly for our work, we had ground control points spread throughout the field that were visible. And I'll talk a lot more about ground control points, or GCPs, in upcoming slides. For image processing, we use Agisoft Metashape, and our UTM zone is 17N. So that's how we export all of our images and handle all of our data. That's where we are in Ontario. We use positions estimated from our ground control points. And in the end, all of our images had under 5 centimeter ground sample distance for our images. So a really key point is aligning those images. So your images need to be georeferenced. So this would be RTK or PPK, so real-time kinematic or post-processing kinematic level accuracy. So your kind of centimeter level accuracy for your images. And there's a few methods to do this. So in the paper, we describe using ground control points, or GCPs. So what these are is essentially a target that you would put out in your field. So whether that is a patio tile like we use, you can see it's a white patio tile there, about 3 by 4, I think. You can get specific drone targets. You can get a number of different shapes. It depends what you want to use or what you have access to. For us, it was easiest to get patio tiles. And the idea is that you put these out throughout your field so that they're visible from your aerial image, and then you get a very accurate position of those ground control points. So that would be using an RTK type GPS, RTK GPS equipment to get those positions. And recently, there have actually been some advancements in UAVs so that we can actually get georeferenced images directly from our drone equipment. So an example would be the DJI P4 multispectral, and that has built-in network RTK. I'll talk a little bit more about that, as well as a newer method of direct georeferencing. And this would be, for example, a drone equipped with an Aplanix APX15 IMU and receiver. And I'll talk about why those would be beneficial. So, you know, in 2018 and 2019, we really used GCPs to get our accurate georeferenced images. But in late 2019 and into 2020, we actually changed some of our methodology. So this is a little bit beyond what was in the paper, but we can now get drone hardware that writes network RTK position metadata right into the image captures. So you can do this as via direct injection of network RTK. So this is, for example, how the DJI P4 RTK works. You can get an ArduSimple RTK for a MicaSense camera. And what these do is essentially write the position of the camera at the time that the image is captured. You can overwrite your metadata from a drone flight using PPK, so like Cloud PPK software. And like the Aplanix IMU, you can overwrite your metadata post-flight with the direct georeference software. So Aplanix has a system to do that. And essentially the difference with the Aplanix versus how DJI does it is that with the direct network injection, we can get position of the drone, but when we use a direct georeference, we get not only the drone position, but we get the orientation of the camera. So rather than something like Agisoft Metashape solving for position of the drone and angle of the camera, we don't have to solve that. We now know what those numbers are. So this makes it really easy to import our images to Agisoft Metashape, which is what we use for our orthomosaic generation. And we can get a georeferenced orthomosaic as an output. The nice thing is that this removes the need for ground control points and their positioning while we can still maintain our centimeter level accuracy of our images. And GCPs are useful, but they do pose some problems in the field. So if you have them spread out throughout a field and you record their position, well, if it rains and then a field crew comes along and drives some heavy equipment over those GCPs, they're going to move. So you don't want to be having to get those accurate GCP positions every time you take an image, because it's a lot of work if you have a very large field. So there are some big benefits to using some of these newer techniques where you can directly get your image alignment without ground control points. So a bit more about the equipment that we used for this work. So we used the Wintersteiger Plotseed TC, which was kind of the linchpin of why we were able to do this. So it was purchased in 2019, and it came equipped with, or we bought, I guess, to add to it, the Trimble TMX 2050. So this is a network RTK system so that we can do precision planting for the soybean breeding program. And this is an example of some of the output from the planter data table. So this will vary by brand, but these are some of the fields that the Trimble system will output for us. So we get, date. So this was actually from this year. So the planting date, time that the plot was dropped. You can see that that's our, the name would be our up. So the direction that the planter was going and the row that it was doing that in, and as well the accurate position of that plot. There are a number of other fields, and again, this will vary by brand, but what you can do is you can also add fields. So in our case, we can use this planting order and time, and we can join those to get our plot IDs directly into our GIS software. We can also use a grid system based on rows and runs to get our plot IDs in there. And it really depends on the software that you have access to. It's also possible in some cases to put your field plans right into the precision planting equipment interface, and then you have your plot IDs and everything directly associated with those points. So you don't even have to do that step. It just depends on the system that you're using. So the specific method workflow that we used is we checked that all of our equipment was RTK capable. So in our case, our planter was, and we had handheld receivers so that we could get our GCP positions. We obtained RTK positions for our GCPs, and then we imaged our field of interest, in our case with a standard drone with no RTK or anything. We use photogrammetry to mosaic and get those centimeter level georeferenced images with those RTK positions for the GCPs. We overlaid our georeferenced image with plot data from the planter. And then, we go into our QGIS software and we're able to convert those points into polygons using the rectangles, ovals, and diamonds tool. We can assign our plot ID, and we did this using our field plan joined with planting timestamps because our breeding program software allows us to output our planting order. So that made it really easy for us, but there's a number of ways to do that depending on the data available to you. And at that point, we can extract our zonal statistics for any kind of downstream analysis. And most of this can essentially be automated, so you don't really have to worry about the intermediate steps. You can get right to data analysis. So a little bit more about the method itself. So, here on the left is an image overlaid with planter data. So you can see that that's some plots are mature and other plots are currently maturing. So you can see the points, and those would be the plot centers as recorded by our planter. So the data from your planter may require some formatting prior to input. It again depends on the brand of planting equipment that you have and how it's set up. In our case, we were able to directly import the shapefile with those points into our GIS software, and we're ready to go from there. You can also record your plot positions using a handheld GPS device if you really wanted to apply this method. And this would work if you had maybe a smaller field, but if you're up in the thousands, it's really handy to have that precision planting equipment. It makes it a lot easier. So this is just what that QGIS rectangles, ovals, and diamonds tool looks like. So you can set parameters based on your plot dimension. So in our case, we want a rectangle because our plots are rectangular. In practice, our plots are about 1.4 by 5 meters. So, if you want a bit of a buffer, you could lower those width and height measurements a little bit. This will depend on your own field and how that is. And the idea would be to generate a bit of a buffer so that you're only really getting the true center of those plots. And your rotation would again be based on the rotation of your field. And then, on the right, we have our output boundaries. So we can see in the middle, we have those plot points and then the boundaries around them, which inherit the attributes of the underlying points, which is an important aspect of the GIS software. So if we do our joining and get our plot IDs with those points, the boundaries will inherit all of those attributes. So here's what it looks like on a much bigger scale. So in the top left, we have our remote output event. So that's what the planter outputs. You can see that lines up pretty darn well. And then on the top right, we have our plot boundaries, which we've generated from those points. And you can see that there's, in the bottom right, there's a later season image, and it still lines up properly. And if you look around the edges, you can actually see that you can see those white patio tiles throughout the field. So that's how we were able to align all these images, because we can see those patio tiles from all of our images. When we zoom in a little closer, so here's a July 4th, 2019 image and an August 14th, 2019 image. And you can see that the boundaries line up very well. We're not bleeding plot to plot, and we're, within the boundaries of the plot. If we were doing this for research, we'd probably maybe make that 5 meters, maybe 4 or 4.5, just to make sure we're not getting the end of the plot, because this is pre-plot trimming. But that's very easily adjustable as we go through and do the work. So, some strengths and weaknesses. So these precision boundaries are objective. So we're not making any input on our end. So we're just getting the data off the planter, which is centimeter level accurate. And it's fast. So we can, whether we're doing this for 100 plot points or 5,000 plot points on a field, it takes the same amount of time. So the turnaround time is very quick. And in terms of turnaround from the machine, we can just download that shapefile and get a drone image, and we're ready to go very quickly. The only real weaknesses is that you require precision planting equipment. Now, this is becoming more common as the cost of this equipment is starting to drop a little bit. And more breeding programs and research programs are moving towards this type of equipment as they're replacing older equipment. As far as images, you do need a very accurately georeferenced image. So this would be like PPK or RTK georeferenced image, and that is not too expensive to get a handheld receiver that does those types of measurements if you go the ground control point route. And the drone equipment route for newer equipment that can do this direct georeferencing or network RTK injection is dropping in price as well. So with that, it's actually a fairly straightforward and simple method. The nice thing is that these plot boundaries provide a high throughput and objective delineation of plots. And this works, from aerial imagery. It works from satellite imagery too, if you have high enough resolution satellite imagery. It's very easy to integrate into an analysis workflow. So the underlying scripts in QGIS, I believe, are all Python based. So you could do this all command line if you really wanted to. And with precision equipment becoming a lot more common, some research programs may already have some of this data without even realizing. So it was more that we bought this planter and realized, oh, we can do this with the data. So that was really nice to see. And now we can do this, for all of our locations very easily. So really the bottom line takeaway of everything is if you accurately georeference your data sets, everything else will line up. So you have accurate point data, you have accurate image data, everything will line up. And this is especially important if you're working with, say, time series. So you image a field, 8 or 10 or maybe 20 times throughout the field season. If you have accurate georeferencing for all those images, your analysis downstream becomes very, very easy. So I'll just spend a couple slides just briefly explaining, how we're applying this method and, a few ways that we're doing that. So we've looked at DEMs for plant height. So you can see on the left here, we have a visible image in the top left, and we have the DEM associated with that image or digital elevation model in the bottom left. So the best relationship we found between our measured height and our DEM height was at our full maturity date. This was an October 15th flight from last year. The DEM tends to underestimate height a little bit, but if the DEM predicts it as tall, chances are it's a fairly tall plot, lodging aside. We also got a decent relationship from full canopy cover images. Interestingly, the relationship falls apart if you have mixed maturity. So if you have some plots that are fully mature and some plots that are not mature at all yet, the tops of those not mature plots will appear a bit higher in the DEM, and it does throw off this measurement. So it is a consideration for DEMs, but it's an interesting use of it, and we can potentially automate getting plant height from our field locations. A big one that we're looking at right now is soybean pubescence. So here's some ground pictures of a gray soybean row and a light tawny soybean row and a tawny soybean row. And if you've ever had to rogue soybeans, you know that pubescence is a big challenge to visually assess, especially that light tawny and tawny separation. So we propose a machine learning method to predict pubescence from aerial imagery. I'll explain a little bit more about the details. So first, we actually started off with some ground-based spectral measurements. So we used the Unispec DC reflectance spectrometer. So this is a ground-based handheld machine that has a probe that looks up and gets the sunlight, and then a probe that looks down and gets the reflected sunlight off the canopy. So this data is 344 to 1102 nanometers at about a 3 nanometer spacing. And this is a whole plot scan. So we're getting the mature soybeans, and we're getting any weeds in the ground and any dirt as well. But when we look at that full spectral data for 60 plots where we know the pubescence, we see that there's a bit of a separation there between our gray and light tawny and tawny. So that's really interesting, and looks good that we can use some imagery to differentiate these. But then we asked, well, what happens if we don't have this hyperspectral data? What happens if we go and look at just the bands that simulate what a typical drone camera, which is a 5-band red, green, blue, NIR, and red edge might have? So when we pulled those bands out of our Unispec data, we actually get, again, a very good separation of our classes by the looks of it. And we're still accounting for essentially 98% of the variation in those first two components. So this is looking really promising. So what does it look like for the drone imagery? Well, actually, we see a very similar trend. So this is from a training data set that we have for our soybean plots. And again, we see the same association, or the same amount of variance captured in those first two PCs. We see a difference between our gray, light tawny, and tawny classes. And as well, we see, and we see on PC2 and PC3, we see that, the gray is really separating out. So this is looking really promising. So, from an aerial image, we can see that there are definitely, visible differences, and these subtle differences we're able to pick up. And ideally, we have a model that we can train on a given data set and apply to many images. And hopefully this workflow is fast compared to manual phenotyping, because this is a challenge to do this on a breeding program scale. So for this, we used a support vector machine classifier. And here's a bit of an example of the workflow of that. So we take our original image, and there's our precision boundaries that we've drawn in order to figure out what is what in the image. We did a no soil, so we removed all the soil pixels, and then we end up with our plant pixels there. So these would be representative of the actual plants and the color of those. And then this is an older version of the model, but you can see that we see differences between our tawny, our gray, and our light tawny. So the idea is that we can predict these on a wide scale. And this is work that we're kind of just finishing up now, and it's looking very promising. And one final slide here on a bit of soybean physiology we're doing. So you might notice, or you might have noticed in some of these plots, that there's holes. So why are there holes in the soybean plots? Well, these are part of a multi-year project in collaboration with Dr. Hugh Earle, a crop physiologist here at the University of Guelph. So we're looking at changes in soybean development from our aerial imagery. So he's doing this all on the ground and with the breeding program on a set of traits. And we said, well, let's have a look at this from the aerial imagery and see what we can do. This is a bit of an add-on to the project. So, we're looking at a few traits, plant biomass, growth rate, and whether we can, get a good final yield estimation for those. So, this is something that we're working on right now, and we're really hopeful that we have a lot of really good ground data and that we can get some good analysis done and figure out what's happening from the aerial imagery side. So with that, I'd like to say, thanks for attending. A brief note on funding. So this was partly funded from Canada First Research Excellent Fund through the Food from Thought Agricultural Systems for a Healthy Planet, as well as NSERC Collaborative Research and Development Grant. And our, industry partners for that include CanGrow Genetics and Huron Commodities, as well as the acknowledgements from our paper. So that'd be Corey Schilling for help on the soybean breeding side and the equipment, and Larry Prong for help with the Trimble unit to get some of the useful data out of that. And of course, thank you to the Plant Phenome Journal for inviting us today, and we're happy to take any questions. So thank you very much.
Okay, well, thank you very much, Dr. Bruce. So I'm going to open it up for questions here. Please feel free to drop something in the chat. I'm going to ask the first question while we're waiting for things to show up. And then if we don't get a lot of questions in the chat, we'll open it up to some audio or oral questions. So one of the things I'm sort of curious about is it looks like you've based everything based on the planter. In our experience, sometimes where the plot boundaries don't line up are later in the season due to mosaicing issues. Have you experienced any of that?
So from the soybean images, I haven't seen any big mosaicing issues. John could maybe provide a better comment because he's looked at a lot more imagery than I have, but I haven't noticed that as an issue with our soybean work. John, any comment on that?
Yeah. Basically, I'd have to look at what your processing steps are in your mosaicing workflow. But what GPS are you using? What type of GPS are you using?
Well, Scott's on. I think he could probably answer that better than I could at this point. But you know, the 2017 data was the data set that I remember we had or 2018 data we had the biggest problem with, and part of it had to do with not enough overlap for enough tie points to do a good mosaic, and so then that translates. Yeah.
Okay, there's even when you have that type of setting, you can often go in and manually add some key tie points, and that will help out. So there's some manual steps that you can take to improve that. But yeah, if you don't have enough side across track overlap, enough side lap, then the software will require, it won't give you a good enough result, or else you'll have to do some manual tie point editing so it can try to match those. And then the other thing I asked about the GPS is basically if you're using a GPS without any sort of corrections, the accuracy could be anywhere from 5 to 8 meters, right? And when the software is looking at these individual captures, it's trying to guess at what their positions are. And if you're just using the stock receiver on something, for instance, a MicaSense, that is only using GPS satellites. It's not using any other constellations, and it's not using any sort of corrections. So you're not going to get great results right off the bat from doing photogrammetry with uncorrected data than you would with a higher accuracy in the GPS. But anyway, to salvage that data, I would just try to go in and manually add some tie points in.
Okay, thanks.
Yeah.
We've got a few questions here. First from Luciano Sharatucci. How do you deal with orthomosaic errors? Do we need to use RTK drones only? So sort of a similar question.
I'll answer that. It all boils down to which specific types of errors you're getting. And so it's probably worth, I know that Pix4D does a lot of trainings and Agisoft has a lot of training. It's probably worth investing the time on learning how to go through and set up a project and figure out how to troubleshoot and adjust your settings in case you're not getting a result that you like. I found even with a low accuracy GPS, I can get very high repeatability by using these ground control points. So it's not absolutely necessary. It just makes it a lot easier on the software from the get-go, because if you're using a GPS with 5 meter accuracy, it's going to have to translate all those points 5 meters or so, or guess how much it should. Whereas if you start off with even something that had 1 meter accuracy, it's going to be a lot easier for it to finally figure out what's the right solution on that as a starting point. So it's not strictly necessary, but it helps a lot. But with your processing issues, I'd have to see exactly what the output is, and then you probably just have to iterate through a couple of different settings that you modify in when you do the tie point matching, or and it boils down to the software too. If you're not using a software like Agisoft or Pix4D where you can go in and throw out bad image captures and edit stuff, if you're just uploading to a service, a lot of times when you're uploading to one of these cloud services, they're not going to be as conscientious about throwing out images with shadows, cloud shadows, and things like that. So you just won't have as much control there. So it really kind of depends on, are you doing the processing yourself or outsourcing it? If you're doing it yourself, just check through some of the settings and modifying those so you can get through a better result.
Okay, great. Kevin Wang asks, thanks to the presenter, very nice presentation. One question about the RTK GNSS. Does the RTK receiver on the UAV share the same RTK base as the RTK receiver on the planter?
So the base in both of those was a virtual reference station. So what that was is a simulated base station. And so, of course, the planting and the drone imaging were done at different times. So they weren't getting the exact same baseline corrections, right? But what they were getting was simulated baseline corrections from a virtual reference stations, which is, I think I have some references in there for what a VRS is. But so the answer is, well, yes, if you think of virtual reference stations the same baseline.
Oh, I'm muted, sorry. Scott Wild says, responding to our previous discussion in my program, we were using GNSS. We also have an RTK system this year. I'll also point out, if you ever do have any interest in looking at corn data, all of our 2017, 2018, and I think 2019 data is publicly available, like 18 flights on 5 acres of plots on Cyverse.
Wait, where's that at?
I'll be happy to send you a link. It's on Cyverse. We made it publicly available a couple of years ago, or the 2017 data. Now Scott's got up the 2018 and 2019 data, I believe. I'm
doing a lot of on-farm research with corn right now. And so if there's some other like experimental data out there that I might be able to use, then yeah, I'll definitely dig into it. And I appreciate that offer. Sure.
So that, but he also has a question here. Great talk. Could someone use their planter without a GPS system? Could they just mount it with an RTK system, basically using a handheld mounted on it?
No. You've got to have, I guess it kind of depends what you want to get out of it and how many attributes you expect to be in the shapefile. So if you did like a prescription planting and you had like the variety, there's a lot of stuff that we didn't have on in there that we had to manually join back in later. But if you're just using an independent GPS, it'll just be that ideally the center of the plot. But the way that the planter works is, which we listed this in the paper, we described this, wherever the antenna is, and so the antenna would look something like this. It has something called a phase center, which is somewhere, it kind of varies, but what it'll do is it's going to log wherever the center of that antenna is. And for us, the way the planter was set up, that was at the midpoint of each plot. And then that GPS reading was triggered by the planter actuating and drilling the seed in. So if you had a GPS that was independent of the planter, I'm not quite sure how you would trigger it and make sure that remote output event, coincides with the same timing as the GPS antenna, or the GNSS antenna. So one thing I was saying could work is you could just have a very low cost GPS, like one of these RG Simples, which cost a few $100, and you could just put it on a post and go out there and you can get the midpoint of all your plots and just walk it. And that would take a horrible amount of time. Go ahead.
Oh yeah, one other option. I mean, if you have an older style planter, you can always use like an RTK type GPS to line up your baseline. And that would at least help you line up your plots. But yeah, it doesn't necessarily help you get the plot centers unless you're going to go and manually do that.
Yeah. But there might be a way to hack it.
Istvan, you go ahead. I mean, this is an open discussion.
Yeah. Thank you, Seth. I just wanted to add that since this is happening in my program and we purchased the precision planter last year, I can't emphasize enough how much of a difference this has made to our program. And basically, because of the way that the GPS planter works, it plants the plots so precisely that we had never seen plots lined up as well as they are now with the precision planter. So having a handheld RTK that's not coordinated with the way the planter is working might not be ideal. It requires the coordination between the 2 points, as I understand it.
Okay.
Thank you.
Thanks.
Thanks, Ishvan.
You're welcome.
So Dale Wilson, I see he's still on, but I'll read. He's analyzing data from an affiliated company. He's seeing a very strange error distribution from NDVI plot data, about 2/3 very tight data and about 1/3 huge outliers. I'm not sure what the question is there, but maybe you can interpret that. Oh, he's on.
Oh, hi. Thanks for this seminar just came up out of the blue for me. I'm basically a user of these data. Here, let me tip that down a little bit. I'm basically a user of these data, and to me, it's like a black box. But there's some kind of systematic problem going on, and I don't even know the right questions to ask. I think that, I mean, this is UAV data collected by a vendor, and I haven't been involved in the details of the process. But I wondered if this is, is this a common kind of occurrence? And B, how would I proceed to troubleshoot this?
So I'd have to see what that looks like. Do you have any graphical output showing these data distributions or maps or?
Well, I got one. I got something up right now. I could probably share. I don't know. I might mess up your talk if I try to share my screen.
That's okay.
But yeah, I'll give it a try. Yeah, I was just analyzing this in SAS. If I can find where
It's either the box at the top or the box at the bottom. It disappears when you don't have your cursor over it.
oh, go show all the windows. Okay. Yeah. I don't know.
While Dale's pulling that up, I'll just, it's not a question, but Godwin said, very interesting, sounds exciting. Though unfamiliar technology where I come from, he wishes he could learn more about this. And thank you for your presentation.
Oh, thank you very much. There's some really good online courses from, I think it's Penn State on GPS. I can pull up a link to that if you want, if you've got the time to learn on your own. They've got some free teaching resources.
Guys, a good place to drop that is in the chat if you get it, if you find the link for it.
Okay.
Okay. Do you see the screen now?
We see SAS output.
Yeah, I got SAS output. This is just what I was working on late last week. Oops. And this is just an example of NDVI data. So you know, I'm using Huber's M estimation to parse for outliers. These kinds of outliers would blow OLS estimation kind of out of the water. But so a lot of these data are very tight, very small errors, but then some of them are like 30 or 40 standard deviations away from the expected value. It's like something went very, very wrong in this, I think.
Well, there's lots of opportunities for things to go wrong between when the data is collected and when it ends up in an output like this. So what I would first ask is what type of plot crop it is and what development stage.
Well, this is from 100 days after planting in central Illinois.
Mm-hmm.
So these are big corn plants.
Oh, okay. So this is corn, and you're
Corn,
measuring
yeah.
a plot, and the dimensions of each of these observations, it's the same
Well,
size
the
plot?
plots are 10 feet wide and 75 feet long.
Okay, and so what's happening is, did you do some sort of statistical analysis with that NDVI?
Oh, yeah. Well, this
Okay,
is.
so what do you relate it to?
So what I do is I fit a preliminary model using proc robust reg and SAS, and then you use some robust method to look for outliers. With such big plots, I mean, the experiment was very nice and we got significant yield results. And the yield results are correlated with the NDVI results. but only after I remove these terrible outliers. It's
Right.
very interesting data.
Yeah. Okay. So the outliers. So this is what is that residual of?
Oh, those are standard deviations.
So that's between NDVI and there's some difference.
So in other words, this 37 standardized residual here from this particular plot, that was the observation was 37 standard deviations away from where it should have been based on the other data in the trial.
So it was higher yield predicted than observed?
Yeah, that was 37 standard deviations too high. Well, I mean, this is not yield, this is NDVI here.
So it's just, okay, so how far it is. So what I would do to troubleshoot this is, this looks like some sort of univariate type of residual.
Well,
Like,
so this is just the univariate based on the 100-day NDVI.
well, so how I would troubleshoot this is regress yield onto NDVI, and then look at your errors, and then go back onto the red, green, blue display, not the NDVI map, and look at those plots and see if the plot boundaries look right, or see if there were like if there was poor stand establishment there, or if there was some other sort of damage that impacted it. But the thing to do is go back in and look at those particular plots and say, does this make sense? Because you could have one plot that does just do horribly worse than the others, and then you get a large negative residual.
Oh yeah.
That could just
Well,
be
yeah.
because
See, I don't see these residuals aren't correlated with yield residuals.
Yeah. Well, so but it only matters when you try to predict some biophysical attribute with it,
Yeah.
right? Because the
I think there was
NDVI
some-
on its own doesn't mean anything.
Yeah, I know. It looks. My guess is that these are some the plots were not justified correctly, or they weren't. The boundaries were not.
It could have been with something that our paper addressed, which is, does your data line up? So if your data doesn't line up, you can get weird things like this happening. But what I would do is, whatever you want to use NDVI as a proxy for, or for NDVI to represent, relate that through some model, and then look at that model's errors and map out the errors. You want to look at a map of those errors.
Yeah,
and then
I also have multiple NDVI readings every 2 weeks. I haven't.
Well, yeah. So what
I
you can do
haven't
then,
done this over time yet.
per plot, what you could do is graph that plot's NDVI progression. And then here's something else. Are you using a red NDVI or a green NDVI, or which NDVI is it?
Boy, I sure don't know. They just.
Well, I'll tell you
sent me
something with
NDVI.
corn that I've been noticed since I started working with corn, was it last year? Green NDVI will, from my experience, always vastly outperforms red NDVI. And there's some reasons for that. Partly because the corn leaf is so dark in the red band that it saturates and you lose sensitivity. And when you put that into that index, the green NDVI, I find at much earlier development stages, and just about any development stage, as I compare green NDVI to a host of other indices. I have a presentation on this for the sensor-based nutrient management community for the upcoming ASA meeting that would have been in Phoenix, where I talk about this, of just if we're trying to predict yield with corn, which indices work better. And but anyway, what I would switch to using, if that sensor has a green channel on it, I would calculate NDVI with a green and infrared instead of red and infrared.
Okay, really good. I won't take up more of the time here, but this has been very helpful. I would know. Let me see if I can.
I can stop
sharing
here.
Okay, go ahead.
Yeah, just a couple other points that I guess we've experienced. So plot boundaries is a big one, but you should be able to see across all of your flights if it's plot boundaries. The other thing at 100 days that we might see here is differential and drying down. So you might have dry plots versus green plots, and that would cause a big difference. Or the tassels, or also weeds. So it may be actually a real difference, but the only way you can see that is if you actually have the images. And it sounds like you just were given tabular data.
Right. Yeah, I haven't seen the images yet, but I want to see them because to better understand what's going on.
Maybe Seth, if I can just contribute a little bit to this conversation here, is that based on that huge standard deviation, it looks like images might have been saturated. So like it looks like a 37 looks like an old NDVI value is equal to 1. Negative 37 might be NDVI value is negative 1. So images might have been saturated. So I'm not sure. There's a lot of places that can go wrong. Like, and have you done, radiometric calibration correctly? What kind of sensor you use? A lot of times, like a vendor claim that they calculate NDVI, but sometimes they claim that they can calculate NDVI using RGB sensors, which is not true. So I think with this, certain information that we're looking at, it's very hard to tell what went wrong. But to me, just looking at this in a table here, looks like the data collection, maybe the sensor was not correctly configured, and the sensor ended up in a saturated measurement out of those.
I see. Yeah.
You just need to map your errors. If you don't have a facility to map your errors, it'll be tricky to troubleshoot them.
Well, I think this is a new first for the Plant Phenome Journal webinar where we get people sharing their results and diagnosing them, but I guess it sounds like it's fairly helpful for people. I'm not seeing any other questions, so if anybody else has any questions for our speakers or for any of their collaborators, I'd invite you to just unmute and go ahead and ask them now.
Okay, let me see if I can.
And hearing none at the moment, I'll ask one last question here, which is, John, you pointed out that your background is geography. You didn't have a lot of agricultural background. Robert, on the other hand, didn't sound like he had a lot of remote sensing background, maybe more on the genetic side. So you two have been collaborating, and then you got Istvan also, who's on the agricultural side. So I guess what's been the biggest challenge for you guys working together, and how have you sort of overcome it? Because I think the transdisciplinary aspect is always one of the biggest challenges, the personnel, the personal interactions as opposed to the science.
I mean, I guess I can start off there, and I'll say that, from my end, it was kind of learning the language of remote sensing and, even the basics of the software. Like there's always a bit of a learning curve there. And I actually took John's 4th year precision agriculture introduction class, and, that gave me a lot of the basics. And, that really helped me figure out, hey, we can really apply this to our plant breeding questions and get some really good data that's useful for us as plant breeders and geneticists and researchers.
Yeah, and I'll go on to just add that it was a lot easier because Rob is one of those people who you can explain something to and ask him to do something, and he gets it, and he just does it. And that makes it a lot easier. But we didn't have any, but a lot of that's just because of his aptitude, I'd say, is why it's actually been pretty seamless and ideal for me. But yeah, what you do need to do is take at least one course where you learn some of the foundations of the concepts, and then you just build on those and apply them to whatever you're interested in. And then a lot of stuff should fall into place.
And thank you. And for my part, I'm a soybean breeder at the University of Guelph, and all these images were taken in that are part of the paper, and also additional discussion that Rob offered are part of my breeding program. So how I got involved is I had Rob as a postdoc until May of this year on the mentioned project that's funded by NSERC CRD. That's the National Science and Engineering Research Council of Canada Collaborative Research and Development Program. So Rob started talking to John independently as part of the project, and we came up with ideas that we had in common, and trying to start something new that would benefit our program as well as potentially other soybean breeding programs elsewhere, and potentially other crops as well. So I'm pleased to see the attendance and also contributions from researchers who are working on different crops other than soybean being interested in our results. So I appreciate it. Thank you.
Right.
Well, thank you all. Any last chance for questions? I know sometimes people are shy. Oh, we got another chat. Oh, okay. Well, hearing none, thank you for the speaker and the colleagues and everybody's attendance. We'll look forward to seeing you next month, first Monday of the month, for the next Plant Phenome Journal webinar. Have a great week.
Thank you.
Thank you.
Thanks.
Thanks.